ill-defined aspect of secretion, but they turn up almost as highly represented as DNA JC5 in this analysis. The scamps are actually representatives of a family of 5 genes, scamps 1 through 5, only 3 of which turn up because these 3 have a significant N-terminal cytoplasmic domain that could be targeted for bioassimilation. So here is a representation, a simple representation of scamps 1 to 3 with this N-terminal domain. Scamps 4 and 5 don't have such a domain. Scamp 5 is a neuronal specific form of the set of paralogs. Just to summarize, the function of these proteins has remained elusive. They're known to be quite abundant. They're present in cells in micromolar concentrations, so they're fairly abundant proteins. They're known from proteomic databases to be enriched in endosomes and lysosomes. They are conserved across all higher eukaryotes, animals and plants, not unfortunately in yeast, which would have made their analysis simpler. And scamps 1 and 2 and 3 are quite abundant. Scamps 4 and 5 are less abundant. So we've done 2 experiments, one of which is shown here, that suggest that these scamps may have a role in the unconventional secretion of alpha-synuclein. Shenji has found that when the scamps 2, 3, 4, or 5 are overexpressed in just standard cultured cells in which alpha-synuclein is overexpressed, there was a substantial increase, perhaps 8-fold increase, in the unconventional secretion of alpha-synuclein. Scamp 1 is already so abundant that it's difficult to overexpress it, so that seems to be less important here. In contrast, if we knock down each of the scamps individually, there was no effect on the unconventional secretion. So there's some curious relationship between the balance of these proteins as seen by overexpression or knockdown. Now, it's been very difficult to create multiple knockdowns because of the 5 paralogs. We're still busy doing that. But in the course of our work, we learned of a team, part of this big Aligning Science Across Parkinson's network, that has been investigating Drosophila as a model of Parkinson's. Drosophila, for instance, has the gene LARC2. It does not have the gene for alpha-synuclein, but a team at Harvard Medical School has shown that if you produce human alpha-synuclein localized to the fly brain, there is a motility defect. So here's the team that I'll tell you about. The PI in this team is Mel Feeney, who's a team member in one of our ASAP programs, and her postdoc, Vanitha Nathidanandam. The 2 of them have done the experiments that I'll show you in the next slides. They availed themselves of a published 2-deletion set of fly mutants in which most, but not all, of the single scamp paralog has been deleted. By immunoblot, the protein seems not to be detected at all, but this is because the antibody may only recognize the part that has been deleted. So these mutant flies have been described in the literature. They have a neurologic defect, but there's no evident motility defect associated with the deletion of scamp itself in the fly brain. Now, in contrast, as you'll see here, and which is summarized on the data on the right, the 2 scamp deletions, shown in bars on the far right, show perfect motility at the movement assay, quantified movement assay of flies, similar to that seen in a wild-type fly. The expression of alpha-synuclein in the fly brain, human alpha-synuclein, reduces this motility down to about 30% of normal. But the expression of the human alpha-synuclein in the scamp mutant flies reduced the motility of these flies to paralysis. There's no movement. And this can be seen quite nicely in the images that I've shown of fly vials to the left. So here's a normal fly in its motility, jumping around in this last slide. The scamp mutant, one of the scamp mutants, shows perfectly normal motility. The expression of alpha-synuclein alone, as you see in the lower left, clearly slows motility of these flies. And then finally, on the right, the expression of alpha-synuclein in one of the SCAMP mutants paralyzes the flies so that they have no evident motility at all. So these experiments suggest that the SCAMP, perhaps under conditions where alpha-synuclein is expressed, really play a role somehow in motility. Another piece of evidence that Melfini's lab has produced in my last data slides is shown here. Under conditions where in the normal fly, alpha-synuclein is expressed in the fly brain, one can see an accumulation apparently of aggregates of alpha-synuclein in the cytoplasm of these neuronal cells, which is not seen in the control where there's no alpha-synuclein being expressed. But in contrast, when the alpha-synuclein is expressed in either of the SCAMP mutant flies, there's quite a substantial increase in the accumulation of aggregated forms of alpha-synuclein, as though it can't be processed and possibly secreted in the fly brain. The alpha-synuclein that accumulates is not coincident with markers of the autophagosome membrane, and so we believe, though it's indirectly, the evidence is indirect, that it's accumulating in the cytoplasm. So the final slide summarizes the two broad possibilities to explain our observations. Is that SCAMP may itself be a channel, though I would caution that the predicted structure from AlphaFold models of the 4 paralogs gives no comfort to the idea that it's a channel. It doesn't look like any other protein channel. But nonetheless, it could be a channel that serves as a conduit for the transmission of unfolded forms of alpha-synuclein, which may then be secreted outside of the cell when the endosome fuses. Alternatively, a model that I favor more is that the SCAMPs serve as some kind of a receptor that recognizes either monomeric or aggregated forms of alpha-synuclein, which are then internalized, possibly into a vesicle budding into the endosome, and from which somehow alpha-synuclein escapes across the membrane for reasons that can't yet be explained, which produces a soluble species that is secreted from the cell. So much to do, but some interesting excitement ahead. So let me conclude by presenting the people on the team who've done all the work that I've described. The first experiment that I showed on separation of the two populations of vesicles was conducted by a former graduate student, Roraima Timosh-Diaz. The experiments on a role for plasma membrane damage in the secretion of abundance of extracellular vesicles were conducted by George Renaud and Justin Williams, two very talented graduate students who are still in the lab, and I regret will be moving on to do postdocs this year. Finally, the experiments being conducted on the unconventional secretion of alpha-synuclein were done by a very talented Chinese postdoc, Shenji Wu. So that's it for what I have to say today. Thank you, and I'll look forward to discussion on this subject.
So thanks Dr. Shackman's inspiring talk about the exosome, and I didn't realize the information about Parkinson's disease because it's personally interesting. But I also like to take this opportunity to welcome our Minister of National Science Technology Council, Wu Chengwen, to it. Okay, so I think the time is about right. Randy, if you don't mind, the question, because the whole day that Randy is going to be here. So if anybody who have a question for Dr. Shackman, to save the time that we can leave. Or would you like to take a couple of questions? Okay, I
see a hand over there.
Okay, one question only. Okay, I have a few questions, but I can ask him later on. So please, just one question.
Yeah, very nice to talk, Randy. Yeah, my question regarding the calcium-induced membrane damage. I want to know the polarized cells. Is it uphold or rear end? That's my first question. The second question will be regarding the microdomain of the lace membrane, or even the cortical actin structure. They contribute to this, especially corpine distribution.
Yeah.
Per se. Thank you.
We haven't looked at polarized epithelial cells, but the laser ablation experiments that we've done have been focused on on random specific region of the brain of the cell. And in every instance, we see when you ablate a patch of the cell surface, you get a huge bolus of material being expelled from the cell, and an enrichment of one of the calcium-binding proteins. So my guess is, and the other treatments that we use are very general treatments that would expose both apical and basolateral surfaces on a polarized cell. So my guess is this kind of secretion can be achieved at any position around the cell. And what was the second question?
Hi, Copac distribution.
Copac, yeah. So to begin with, all of these calcium-binding proteins are uniformly distributed in the cytoplasm. And part of this we published, but as soon as you damage the plasma membrane, you can see an enrichment of these proteins that are mobilized to the cell surface, uniformly about the cell surface, but probably when the damage is limited to a particular region, they are mobilized to that very region where calcium is flowing into the cell. This requires millimolar levels of calcium, so the normal calcium flow that elicits synaptic transmission probably isn't enough to cause this release of exosomes.
Okay, so I think I am sure there will be a lot of questions. Actually, I have a few too. So the whole day we have a chance to ask for Dr. Sheckman. Again, thanks to Dr. Sheckman's inspiring talk. Hey, right over here. Okay. Taiwanese is very polite. Always remember to belong. Okay. So before we turn to our minister who is going to give us a few words, because the minister who is very interesting to make Taiwan's technology top in the world. I know that's what your question. And so before that, I think we have something for.
We'll have Professor Hong to deliver a certificate of appreciation to Professor Sheckman.
This is certificate for you. I know you. I know you receive certificate, this kind of certificate, almost every day. But this one, next time when I visit your office, I'm going to check on this one, whether it is hanging in the office or not. So remember that. So if I go to your office, then make sure to find this one. Okay.
Thanks again to Professor Sheckman and Professor Hong. And now let's welcome Professor Chen Wen-wu, the Minister of National Science and Technology Council, to give us the opening speech. We would like to extend our heartfelt thanks to Minister Wu for taking the time to be with us today and for offering his valuable encouragement and guidance to CMU. Under the President Lai's Healthy Taiwan policy, the National Science and Technology Council has played a vital role in leading academia, industry, and research institutions to actively advance innovation in the biotechnology and pharmaceutical sectors. Let us welcome Minister Wu.
Good morning, Professor Randy Wayne Sheckman from Berkeley. The CEO and also Professor Long Bin Jen and President Min-Chi Hong and Dean Yang Chiu and all the distinguished international scholars, experts, scientists, and also students here, and ladies and gentlemen. It is really my privilege and honor to be here today, invited by President Hong. The reason that I'm here as President Hong just mentioned, I'm working very hard to help the academia, the healthcare society, and also the industry to work together, hoping that we can make Taiwan stronger better, and so that Taiwan can help the world. This is very important. So today it is the next generation extracellular vesicle in therapeutics forward. This is very new to me because I'm an engineer. In the past almost 40 years, I have been a professor at in National Tsinghua University in Hsinchu, and I think you can imagine that in Hsinchu, most of the people are working on semiconductors. So in my whole life, I have been working on semiconductor chip design and testing and applications. So I've been working with companies like TSMC, MediaTek, and so many companies in the Central area in my whole life. And most of my students are working in the high-tech companies, and some of them also teaching in the universities. But since last year, I took office in Taipei, and President Lai told me that he has a very great vision of Taiwan. And the national vision is called Healthy Taiwan. Healthy Taiwan is to make the citizens of all age healthy in their whole life. We are not talking about only health care for senior people. We need to improve the health of all generations, all ages, all citizens. This is very important. So I am deeply inspired to see so many international leading scholars and researchers gathered here. So I told President Hong that I must come here and see for myself, to understand what CMU has been working on. And so many talents gathered in this room. That has, it is quite inspiring, and potentially we'll develop something. So in particular, having the opportunity to talk to Professor Sheckman and President Hong is very important for me. As I'm trying, as I mentioned, I'm trying to improve Taiwan's biotechnology R&D support from the government side. As I mentioned, President Lai has this vision of health in Taiwan. And there are 3 directions, including promoting comprehensive health care for all ages, as I mentioned, strengthening integrated medical care, and optimizing environment and innovative development. To do that, technologies like semiconductors and AI and ICT, all the high-tech industries need to collaborate with the healthcare industry and biotech industry all together. So the Bio-Taiwan Committee meeting convened by the Executive Yuan last year strategically planned Taiwan's biomedical industry development blueprint from a national perspective, echoing President Lai's national vision. We aim to promote the integration of biomedical and smart innovative technologies, such as AI and semiconductor ICT, to advance the R&D achievements, expand the overall market, and promote the upgrading of the biomedical industry. As Minister of the National Science and Technology Council, I deeply understand the importance of basic research for biological and technological innovations. The biotechnology and pharmaceutical field is one of the strategic industries we are focusing on. To enhance overall scientific research capabilities, the NSTC strengthens basic research toward international excellency. Based on national technological development needs, cultivating core teams with global research leadership and originality, and core teams like this one, led by President Hong. Particularly in forward-looking fields such as precision medicine, cell therapy, and gene therapy, we hope to combine Taiwan's strengths in semiconductors and ICT with our solid foundation in biomedical research to bring breakthrough innovations to the global biotechnology industry. That's why we have invested greatly in the research team led by President Hong here. Since 2024, the National Science and Technology Council has been funding CMU to implement the T-STAR Center, which is the Advanced Research Center for Cancer. Investing in research on specific cancers and introducing AI tools for new drug development and precision medicine. To further align with international standards, we have also invited Professor Randy to serve as a project advisory committee member. Thank you, Professor Randy. Professor Shackman. This not only strengthens Taiwan's global competitiveness in cancer research and also leverages his expertise, his experiences to provide recommendations for our academic research environment and talent cultivation, helping our country conduct research that makes significant contribution to the society. Facing the changing international geopolitics and various problems in our national society, the academic research teams must be courageous in taking up grand challenges. We must not only develop advanced medical technologies but also ensure their subsequent widespread applications can benefit the human beings. Through today's international forum and scholastic activities, we hope to encourage academic research teams to align with top international scholars to promote our R&D capabilities and translate research results from laboratories to clinical applications, transforming them into actual medical services that truly address people's needs. Finally, I'd like to thank once again all the participating experts and scholars, especially the international guests who have traveled from other parts of the world, for opening new opportunities for Taiwan's academic research. Let us work together to make greater contributions to human health and welfare. I also wish everyone all the best. Thank you.
Thanks again to Minister Wu for your warm and inspiring speech. And now it is time for the group photo. Please remain seated as we take a group photo from the stage. Thank you. Okay, thank you everyone. And we are now ready to begin our next session, the global trends. The moderator for this session is Professor Zhiyang Cho, the Superintendent of China Medical University Hospital. Professor Cho is a renowned expert in neurosurgery, immunotherapy, and extracellular vesicles, with extensive contributions to these fields. His leadership and expertise has been instrumental in advancing both medical research and clinical practice. Let us welcome our Superintendent, Professor Cho.
Good morning, everyone. I'm Superintendent Yang Zhou. Thank you for your attendance this meeting. And now I introduce the next speaker. Next speaker is our great honor to invite him to our Taiwan. This time is the first visitor in Taiwan. He is now is the president of the International Society of Several Vesicles, ISEB president. He is a very great expert for the EB. We have a talk about the EB. He is come from the professor molecular competitive pathobiology of University and neurosurgery of Johns Hopkins. He is also the member of Rich Man Family this foundation for Precision Medicine Center of Excellence in Alzheimer's disease. His expertise is EV production and application. Apart from here, he is a top one highly scientific researcher in the world. His special contribution award in the ICEB, also the distinguished investigator, the ISEB. He is also the editor of Journal of CEB and the cytotherapy. Right now, he is very expert in separate and the EVs and how to produce the EV manufacturing is very important in this talk. About the EV, this hype is a dream over the hot topic over the just the hope in the future. He gives us our answers in this talk. His talk is extracellular physical biodistribution and pharmacokinetics and immunogenicity. We give a great talk to welcome our special talk speaker, Kenneth Reader. Professor, please. Thank you.
Well, thank you so much for that warm welcome. As president of the International Society for EVs, I thought that my job was pretty hard. But this morning I have an even harder task, and that is to follow Professor Sheckman and Minister Wu. so I hope that I can still convey some useful information to you during my talk. I want to start out with, what do we know about EVs today? What are some of the opportunities and the challenges? And, as we heard earlier, every field in biomedical research and applications goes through ups and downs. It has its own opportunities and challenges, and EVs are no different from that. So, I think that we have some long-standing myths in the field that we're starting to dispel a little bit, including the idea that only exosomes are important, right? I mean, there are other types of extracellular vesicles too that can also be extremely important in therapies. We've also found that EVs, although we've often assumed that they are perfect delivery vehicles, we heard from Professor Sheckman that sometimes it's very difficult to actually get their contents into cells, and I'll talk a little bit more about that later. Also, the loading of EVs, how easy is it to load them? How can we load them better? And then finally, one of the things that I'm going to tell you about today is some of our work on immunogenicity of EVs. We have for a very long time assumed that EVs are non-immunogenic, and that may not be the case in every scenario. We also know now, due to the work of David Leiden and Bob Coffey and some other folks, that there are many other extracellular particles. We heard from Randy about unconventional secretion. There are many types of extracellular particles that are not EVs that may also be important and that we may not want to ignore in our search for therapies. And then finally, I think that we've been challenged too in the field with a lot of assumptions about separation methods for EVs and characterization methods for EVs. But together with our friendships, our international collaborations, I think we can solve all of these problems. And we're doing that right now. So I just want to show this picture at the very beginning. This is from it's an illustration from a review by the former president of ICEB, Edith Buzesh. And in this review in Nature Reviews Immunology, she showed us some of this diversity. So EVs are not just one thing. They're actually quite diverse in their biogenesis. There are many ways that an EV can be formed, both from the plasma membrane and its extensions, and from the internal compartments of the cell. And this includes some of my favorite EVs, which are envelope viruses. So in my view, an envelope virus is an EV, and in fact, I come from a virology background, so I have done quite a bit of work in my career on HIV and related retroviruses, and I'm very interested in their relationship with the host EVs. All right, so one thing that I said in my abstract was this. Before an EV medicine can be used in a human, we must first understand and possibly adjust the biodistribution, pharmacokinetics, and immunogenicity. And I want to add to this that knowing the mode of action might help as well. Now, an EV is a little bit more complicated, I think, than an antibody drug or a small molecule drug because the EV itself is a platform. There are many things on the surface and on the interior of the EV that might be contributing to an effect that we see in the clinic. And thus it is much more complicated in many cases than other kinds of therapies. But knowing the mode of action and being able to adjust accordingly is also quite important. So my lab some years ago, we wanted to ask the question of where EVs go in the body. And specifically, we wanted to know, am I different from a mouse? Because there have been many very good biodistribution studies that have been done in rodents, mostly mice and rats. But there haven't been so many that have been done in other organisms. And I'm a part of a department at Johns Hopkins called Molecular and Comparative Pathobiology. We've been working with non-human primate models and other large animal models, pigs, sheep, and so on, since the 1970s. And we've done a lot of work on retroviral infection, its effects on central nervous system disease, and so on. So we have access to some models that not everybody has access to, including non-human primates. And I just want to show one example. So there are many ways in which we're different from a rodent. One of those is the olfactory bulb. So in a mouse, the olfactory bulb is practically like a little mini brain, a second brain. But as you look across species, we get up to humans, and we have several orders of magnitude less volume, less mass of the olfactory bulb than a mouse does. So that's a huge difference, especially if we're talking about intranasal delivery of some drug or perhaps an EV. So that's just one example of the differences there. So to be able to do work, though, in a large animal model, we need to have very sensitive reporters. And so in some of the work that I'm going to be showing you here, we are using a dual reporter. This is from Charles Lai's lab just up the road. And their lab constructed this reporter that consists of an enhanced green fluorescent protein that's linked to nanoluciferase. We heard about nanoluciferase in Randy's talk as well. It's very bright. We can achieve very high sensitivity with this reporter. I'm also going to be showing you some data that has come from another reporter, and this is called MEMFlow. So this is a membrane dye. And in general, we have to be very careful about membrane dyes for a few reasons. One of those reasons is that they can be promiscuous. They can go from one membrane to another without actual fusion. Another reason, and the important reason here, is that lipid dyes can self-aggregate. And so if we mix the lipid dyes with our EV sample, we might see a bunch of particles that are fluorescent, but they aren't actually EVs. They look like EVs by most of the techniques that we use, but they're not actually EVs. And so we don't want to draw wrong conclusions. We're convinced that this MEMFlow reporter is pretty good about not fluorescing when it self-aggregates, and that's because it self-quenches. So when we started this project, Tom Dredongs, who was a postdoc in my lab at the time, and also Ling Lai Zhang was helping with this project, they did some sensitivity assays where they made some EVs, and I'm not going to go into this in a lot of detail, but we basically followed a very standard protocol for producing our EVs in 293T cells, specifically in a line that was suspension adapted for scaling. And then we did a tangential flow filtration and size exclusion chromatography-based protocol for separating those EVs. We did all of the testing to show that they were EVs and that they weren't contaminated with a lot of free protein. When Tom spiked these EVs into healthy macaque plasma, we were able to get several orders of magnitude more sensitivity than we had ever gotten before. In fact, our limit of detection was down to around 20 EVs per microliter. Of course, we were putting multiple microliters into this assay, but it was very sensitive. Now, our in vivo study setup, and since this has been published, I'm not going to go through the entire study here, but we were interested in intranasal exposure, which I'm not going to talk about today, and intravenous exposure, which I will. So our study setup was that we would give the EVs, for today's purposes, intravenously into these animals and do a very intensive sampling regimen for the first hour, where we were taking cerebrospinal fluid and we were also taking blood samples and monitoring what was in them. Then we also had several other time points over 24 hours so that we could monitor the decline of the EVs in different bodily compartments. We also did this with a dose escalation design, where we started out with 7 e to the 10 EVs per animal, and we went all the way up to 9 e to the 12 EVs, and we would administer these doses every 2 weeks. When we had administered the 4th dose, though, we waited another 2 weeks and administered it again. So some of the results I'd like to share with you here. We were able to achieve some crossing of these EVs across the blood CSF barrier in our macaques. And in plasma, down here on the lower left, as you would expect, there was a strong dose response. So the more EVs that we put in, the more signal there was at the beginning. Now, interestingly, over on the lower right, here we are plotting the half-life of these EVs. And in rodent models, and there's been hundreds of studies that have been done, if you look across all of the studies that have been done in mouse and rats, the average half-life of EVs in the circulation is between 5 and 10 minutes. Now that's a little bit daunting, right? Because if we put these EVs into humans, we spent a few million to create the EV dose, we put it into the patient, and it's gone within a few minutes. Well, we found that at least in the beginning, our EVs were sticking around longer in the plasma. They had a half-life of more like 30 to 40 minutes in these non-human primates. I'm going to talk more about this later, though, because that didn't persist indefinitely. Now, what about distribution in different organs? So those of you who have done biodistribution studies in rodents, you know that most of the EVs tend to go to the liver. There are some other places that they can also appear, like in spleen, in lung, and we saw the same thing in the non-human primate. Again, I'm going to come back to this a little bit later, because this is a challenge. If you want to treat a liver disease, that's great. Your EVs are going to go there. But if you want to treat another organ, maybe there has to be an adjustment that is made to the biodistribution of the EVs. Now, where do this was solid organs, but what about in the blood itself? We expected, based on the literature and based upon our own assumptions, that we would see a lot of EV uptake into monocyte lineage cells. And indeed, when we looked at the monocyte lineage cells here in the top row, there was a decent amount of uptake. Or actually, I'm using the word uptake. I want to back away from that. Let's say association, because in the assays that we did here with flow cytometry, we can't say for sure whether the EV is on the surface of the cell or has it internalized. So I'm going to say association. I'm going to try to say association as much as I can. Now, there was association with the monocyte lineage cells, as expected. We also saw some degree of association with a broad T cell population. But the big surprise for us was that our EVs in the non-human primates were primarily going to B cells. They were associating with these B cells, again, a broad population of B cells. And this was something that we were not necessarily expecting. Since our paper came out a couple of years ago, there have been a few other groups that have reported this in other models in the B cells. And since this has been published, again, I'm not going to belabor this, but I do also want to point out that since these studies, we have also moved to using ex vivo blood assays. This enables us to get blood from any organism, including humans, and we can do these cell association assays in the laboratory and not necessarily in vivo. And it has turned out that this B-cell association is something that we see across the board with a lot of different EB sources, but there are exceptions. So not every EB source is associated with B cells. So another thing that I want to share with you is our interest in immunogenicity. Are EBs actually non-immunogenic? So in almost every review on EBs, we read this statement: EBs are non-immunogenic. There are even studies that have been done and reported that cross-kingdom EBs can be non-immunogenic. We might be able to take an EB from a plant or a bacterium, inject it into a human as a drug carrier. Is that really the case, or does it depend on special circumstances? And I have to say, I mean, I've done the same thing. So here's a review that I wrote with Kevin Morris and some colleagues, and we said that too. EBs tend to be non-immunogenic, biodegradable, biocompatible. But there are many other ones that have been written about this, and you can probably find hundreds in the literature that state that EBs are non-immunogenic. So let's ask ourselves the question of why do we assume that EBs are non-immunogenic? So one of the things that I get from a lot of people is that they're small. And I think the people who are saying this are not immunologists. And indeed, I think one of the strengths of the EB community is that we have people from a lot of different backgrounds coming together, but we have to talk to each other. It turns out that the optimal particle diameter range to provoke an immune response is between 2 and 200 nanometers. So this is what the whole vaccine community has learned over decades and decades. So EBs are right in the range of where they should be eliciting a response if they're going to. So that's not the thing. The other idea is that EBs are recognized as self. And this may very well be the case if we have EBs coming from an individual being put into that individual, but it's not necessarily the case elsewhere. And I just want to give you an example here. This is from a paper that we published during the pandemic. We made a bacterial, so this was a bacterial EB-based vaccine where we hooked a receptor binding domain from SARS-CoV-2 onto the surface of bacterial EBs, and we injected it into an animal model, specifically the Syrian hamster model. In that model, we were able to protect those animals from the key correlate of disease, which was weight loss. And so in our vaccinated animals, both males and females, but not in the other control groups, we were able to see that decline did not happen. And in some groups, we actually saw an increase in the weight of the animals even after the infection. So here, clearly, we were getting a response to our EBs. These were bacterial EBs. And this was nothing new. In fact, the very first clinical use of EBs that I'm aware of happened all the way back in the 1980s, when in Cuba, there were some scientists who said, well, why don't we, instead of using killed bacteria, let's just take the vesicles, we'll purify the vesicles and we can inject those, because those vesicles have no chance of replicating. And they made this vaccine, worked very well. It was replicated in many countries around the world. Okay, so we know that bacterial EBs, anyway, can elicit a response, and we can even use them as a vaccine. So what about mammalian vesicles? Taking a vesicle from one mammalian cell type and putting it into an organism. Here, in this case, of course, we're using human cells. We're putting them into another organism, but a very closely related organism. And when we got, I told you about the first 3 doses and the long half-life that the EBs had in those animals. When we got to the 4th dose, though, the half-life declined considerably. And it went down to around the level that we would expect from a rodent model. So why was that? It turned out that there was actually an antibody response that was happening. And by the 3rd dose, or at least the 4th dose, depending on the dosing regimen that we used, we were able to observe significant levels of IgG in the plasma. Now, we also looked at some other antibody types, IgM, IgA. There was also a response there. But in any case, we definitely were seeing a response. And since then, we've done some more work. We've looked at some different types of extracellular vesicles that can be put into our animal model. And we've seen that for most of these, you do get a response, even, interestingly, with MSC EVs. So MSC EVs are generally, they are thought to be non-immunogenic, maybe more than other EVs, but in at least 2 animals that we gave the MSC EVs to, we saw a strong response in one, but not the other. So these are very small studies, of course, and we're working with non-human primates, which are a very expensive model. But nevertheless, we believe that some of these results can be interpreted and can be relevant. However, even in these animals that were having an immune response to the EVs, we didn't see any kind of safety concerns. I'm going to go through these slides very, very fast because there's nothing here, I assure you. We're looking at weight, at CBCs, we're looking at a lot of different parameters, kidney function tests, metabolic parameters, serum enzymes, liver function, cytokines. There was nothing that was significant across these animals. So even though we're getting some immunogenicity with these EVs from a closely related organism, it did not translate into any kind of safety concerns for the animals, at least the doses and the dose courses that we were giving. That's not always the case. So here's a cautionary tale that my lab very foolishly embarked on, I think. There was a paper, not a paper, but actually an editorial that came out in a British newspaper some years ago talking about a rat. So this might be the only rat in the world that's ever had a whole editorial written about it. But her name was Seema. She lived so long that she even got her own name. And she had been given, according to a particular biotech company, injections of blood from a cow, sheep, or pig, or actually a fraction of blood from a cow, a sheep, or a pig. It turns out that later there was a preprint that came out, and we know now that it was from a pig. So we were approached by a company and asked, would you be willing to try to replicate these experiments? Because honestly, we'd all like to stop aging, correct? And it would be very easy if we could just take blood from a large animal, get a fraction of it, and put it into meat, so it cannot age. So we did this experiment. But I said, if we're going to do the experiment, we want to do it looking not just at the blood fraction, but also at the EVs in that blood fraction. And so we did this, and this was done in rat. And the first conclusion that we drew was, this is not a good idea. So this basically a transfusion across organisms. It doesn't work very well. So all of the animals in our study, they got the plasma fraction. They died within days or a little over a week. So that was not good. The other thing was that we saw a very fast antibody response, antibody generation to this foreign material. And this, I think in retrospect, this is not a big surprise. These antibodies were coming up about as rapidly as you could possibly expect them to. And then finally, even the animals that were receiving the EVs. So the EV animals, they did not die immediately. They actually did pretty well compared with the plasma fraction, but they did have an antibody response. So I think that there's a lot more to understand about the immunogenicity of EVs, and as we start to go, especially into human populations, we want to test this. We want to test this for any kind of EV, any preparation that we're using, to make sure that we understand the degree of immunogenicity and whether we have to do something to mitigate that. And here are some of the things that can possibly be done. This is a review from Joy Wolfram and colleagues. I wish that I had looked at this. This is really cool. They came up with some nice ideas for reducing immunogenicity and have some nice references there too. So there are potentially ways around this too, even if we do have concerns. All right. So in the last part of my talk, I want to go into what is an old paradigm, but also a new kind of paradigm in the EV field. So we heard from Randy Sheckman about the difficulty sometimes of delivering EV cargo. We heard from him also about how EVs can be used as trash disposal. This is another thing that we always see in the literature, right? Whenever we write a review, one of the first sentences in the review is, we used to think that EVs were trash disposal, now we think they're for communication. Well, trash disposal can be extremely important. That can be maybe one of the most important functions of EVs. And maybe learning how to modulate that is also part of therapeutics development. But between trash disposal and luminal cargo delivery, I think that there's another way, a third way, that EVs might be functioning. Some of you are probably working on it already. But if we look back at when the EV field really exploded, It was about 20 years ago. So 20 years ago, there was a bunch of work done in several labs, and this was the paper that ultimately achieved the highest number of citations. It was published in the highest impact factor journal of about 3 or 4 papers that came out at the same time. And this paper proposed that messenger RNAs and microRNAs can be transferred between cells in extracellular vesicles. Today, this has had almost 15,000 citations, and it's spawned really a whole industry of people who are looking at the transfer of RNA in extracellular vesicles, which I know that Randy is very skeptical about. I also think it's difficult to do. There may be some situations where it does happen, and there may be ways that we can make it happen, but it's not as easy as we might think. So this led to what I think is currently the reigning paradigm of EV activity, and that is that all this stuff in the middle of the EV, the yellow stuff, that's the important thing. That's the nucleic acids, the proteins, whatever cargo we're trying to deliver. And in contrast, the stuff on the surface, or the lipid bilayer itself, or what's on the outside of it, is simply packaging. You know, it's that package that we send to our friend in the mail. It doesn't really matter the packaging itself; it's what's inside it. So this paradigm too has led to the establishment of many different companies. And one of them was Kodiak. I think this company probably had the highest capitalization of any EV company in the world so far. But it went bankrupt about 2 years ago because it was, I think that they did an incredible amount of work at that company. And if you are familiar with their story, you know that they published a lot of high quality papers. They came up with some very nice techniques. Manufacturing, they did a lot of good work, and we can learn a lot from them. But unfortunately, they ran into these same problems of endosomal escape, of loading EVs, and of getting enough material into a recipient cell that it would work. So when they went bankrupt, I think this the story about the antibodies from the monoclonal antibodies from the 1970s going up and down until today is a very big sector. Well, many investors said, these guys failed, so therefore, this is over. We can't do EV therapies. I don't think that that's the case, but I do think that we have to carefully consider that it's difficult to transfer luminal cargo from an EV to a cell. This is a paper that came out a few years ago that I think very nicely illustrates this. Even if you're able to get your RNA into an EV, that doesn't necessarily mean that it will be transferred into the cell. And you don't need to see the numbers, just the colors on here. On the left, you see multiple cell lines, EVs from multiple cell lines that have specific RNAs in them. You put them onto a whole bunch of different recipient cells, there's probably, 12 or 15 there, and there's nothing that is transferred. Practically nothing. On the right, the EVs have been engineered with VSDG, a viral glycoprotein. This is a protein that will enable fusion to occur and endosomal escape to happen. And there, not always, but for some of these microRNAs and for some of these recipient cell, host or source cell pairs, you actually do see some transfer. So this tells us that we can deliver things with EVs, but we might need to have something extra, something that enables us to do endosomal escape. And I'm not saying that this can't happen. I'm not saying that you can't load EVs. I'm not saying that you can't deliver content. This is just a recent paper from my lab. This is a collaboration with Leigh Zheng's group in Guangzhou, where we showed that we can package U6 into EVs, and this happens naturally in HIV infection. So it happens, but I think that there are some other things that we can do. Now, some of you may know, this is a funny story, I was actually a German literature scholar before I got into science. In fact, I even went to graduate school for German literature. And so one of the poets and writers that I studied was this guy named Hugo von Hofmannsthal. And in one of his books he wrote, "Depth lies hidden." Where? On the surface. And many times we think about the surface. The surface is something that's bad. So we talk about somebody being superficial. We talk about beauty being beauty being skin deep, right? It's just on the surface. You have to look inside. Well, sometimes the surface itself can be the message. Sometimes the surface itself can have depth. And this is the concept that we now call the biomolecular corona of the EV. This is not new either. This comes from 25 years ago when people were first starting to put lipid nanoparticles, liposomes, into circulation. And these scientists found that when you inject a nanoparticle into the bloodstream, it immediately becomes coated, specifically with a hard corona. That's proteins that denature and lie down on the surface, and then also a soft corona, so less strong interactions, weaker interactions, but nevertheless creating this kind of cloud of molecules that coat the surface of the nanoparticle. Of course, an EV is a nanoparticle, and an EV can also have a corona on it. So this concept of the biomolecular corona is I think increasingly interesting these days. There has been work in the past, of course, that's used this, and it makes a lot of sense. So Paolo Vergese, one of my colleagues in Italy, he did a calculation, and he found that EVs, I actually think this might be a typo, I think it's actually less than 180 nanometers in diameter, actually have more protein content in and on the membrane than they have inside them. And you could probably say the same thing about other molecule categories as well. Of course, most EVs are smaller than this. So this means that if we want to look at the bioactivity of an extracellular vesicle, a very good place to examine is not just the interior, but also the exterior. And this calculation does not count that loose corona. So I just want to give you a couple of examples of some things that we've done to look at the corona and how it can be active. One of these are the enzymes CD39, CD73. These are found on MSCs, but also on many other kinds of cells. And what they do is they break down ATP, which is inflammatory in the extracellular environment, all the way down to adenosine, which is actually anti-inflammatory. So Martina Fabiano, who was in my lab for a year visiting from Matthias Ostrowski's lab, was able to show by super resolution microscopy that CD39 and CD73 are on the surface of EV. And furthermore, you can have Thank you. Furthermore, you can have some EVs that are even double positives. They might be handing this molecule from one enzyme to another. The more EVs that we put into a solution or into a suspension, the more hydrolysis of the ATP we have. And this leads to EVs being able to dampen inflammation. And so we did some cytokine studies here. We also saw that EVs will promote neutrophil monocyte interactions. So an EV can function even if we don't know anything about the interior, and probably even if there is nothing on the interior, through some of these mechanisms. Another thing that we've done is to try to engineer EVs to get around some of the problems that we have with endosomal escape or with targeting. So Zach Troyer in my lab, he has been using human endogenous retroviral proteins to try to engineer EVs to have specific targeting, to allow them to achieve uptake, and also to bind to RNAs. And I won't go through all of his work, but I just want to share some really interesting results that he had. Namely, that by putting one of these human endogenous retroviral proteins on the surface of his EVs, he was able to slow down that clearance in the bloodstream. So instead of seeing that there was a decline in half-life over time, he actually observed that the half-life, this longer half-life than the non-human primates, was preserved. And maybe just as interestingly, maybe even more interestingly, here what we have is on the left his results from the biodistribution studies, and on the right the results that I showed you before. And now, in contrast with the liver taking up most of the EVs as a solid organ on the right, you see that the kidney has somehow come to the fore and the liver has declined. So this human endogenous retroviral protein has a target, has a receptor that is at high abundance in the kidney. And so this makes a lot of sense from what we know of the biology. What I want to leave with you today is that EV biodistribution is determined by the EV source and maybe by what we put on the EV. EVs also can be immunogenic. We don't want to forget about that. We want to make sure that we test it rigorously in any EV preparation that we want to put into a human. Safety assessment, though, shows us that at least EVs from closely related organisms are not going to cause adverse events, at least as we have administered them. And then finally, the surface of the EV not just the interior, has untapped depths, and we need to continue working with liminal cargo, absolutely. I know many of you are working with that, we are too. It's not that that's not important, but let's not ignore the surface and the corona. So just thank you for your attention. I just want to say a couple of words about the International Society, for those of you who don't know. We just had a 2,000-person meeting in Vienna, Austria. We have our minimal information for studies of EVs. This came out, the third iteration of this came out last year. If you're new to the field, you want to learn more, this is not the worst place to start. It basically will take you through all of the steps that you need to go through for an experiment on extracellular vesicles into even when you're going into model organisms. I run an EV club. This is an online journal club. So we have this usually twice a month, and we have people who present their own papers. So if you would like to join, or if you would like to present a paper that you have, please reach out to me. Let me know about that. And then finally, we have a global EV network, and I know many of you are part of this. So even if you're not a full-time member of ICEB, you can be part of the EV network through the national societies and local groups. So if you have a local group that's not part of this, please also reach out. And we have 2 journals doing really well. Thank you so much again for your attention.
Thank you. Because time is limited, we will postpone the discussion in the panel discussion this afternoon.
Thank you, Professor Whitworth and Professor Cho. And now we will have Professor Cho to deliver a certificate of appreciation to Professor Whitworth. Thanks again to Professor Whitworth and Professor Cho. And now it is time for the coffee break. Coffee and snacks are in the lobby. Meanwhile, we would like to invite all of you to explore the poster presentation showcasing the latest research and findings from our participants. It is a great opportunity to engage with the authors and discuss their work while enjoying the refreshments. Please feedback before 10:50. Thank you.